Hi, everyone. I'm Claire Mitchell-Costas. I'm Harvey Lodish's scientific lab manager, and I've worked in his lab for nearly 20 years, so it's an honor today to be here. It's always intimidating to introduce somebody who is already so well known at the Institute and who is a legend in the field of biology. What most of you likely know is that Harvey Lodish is a founding member of the Whitehead, is the lead author of the molecular cell biology textbook, is a member of the National Academy, and at age 26 came to MIT and was the first person to ever teach cell biology here. He has also been instrumental in founding many biotech companies, including Genzyme, Millennium, and most recently, Rubius. Harvey has always been generous with his time and has spent many years supporting science as president of the American Society of Cell Biology, as chair of the Mass Life Science Council Advisory Board, as member of the Boston Children's Hospital Board member, and as trustee of Kenyon College, as well as serving many other social, educational, and scientific organizations over the years. Therefore, it's pretty obvious that most people would infer that Harvey is hot on science. What people may not know is that Harvey is not just figuratively hot on science, but literally as well. Harvey holds the MIT radioactivity record for using 1 curie of radioactivity in a single experiment.
P32.
What you may not also know is that Harvey is a genuinely lovely guy. He is an amazing mentor and always supportive of his postdocs, graduate students, and gives everyone who works in his lab scientific freedom and to support their growth. He is always available to meet to discuss scientific data or anything else that would enrich the lives of his trainees. It speaks volumes to Harvey as a great scientist and mentor that his first postdoc to receive the Nobel Prize cited Harvey as the #1 reason he became a Nobel laureate. It's an honor to introduce Harvey today. Without further ado, here's Harvey Lodish.
Thank you. Wow. Thanks. This is great. I've given this talk, a different version of it, twice before, and there the audience was older people who were potential donors to Whitehead. And my goal was to convince them to give us money. That's not my goal today. I'm really speaking to the young people in the audience, particularly the students, the postdocs, the young faculty, who hopefully some of you in future years will be in a position to start and nurture and develop a leading biomedical research institute. And what I'm going to focus on today are scientists who are visionaries, who can see the future, who can build institutions, develop and lead institutions, and convince other people to follow them. I'll also talk about several philanthropists, very wealthy individuals who are not scientists but understand science and are willing to give large sums of money to develop and nurture major scientific institutions so that research can go forward. And at the end, I'll point out how important philanthropy actually is to an institute like Whitehead and similar institutions. But let's start at the beginning. Many of you know when I first meet someone, I'll often ask them, tell me, where did you grow up and how did you wind up at MIT or Whitehead, okay? So let me tell you where I grew up and how I wound up at Whitehead, okay? I grew up in Cleveland in a very tight conservative Jewish community. I was always a very good student, and it was always assumed somehow or other, without telling me, that I should become a doctor. I was a bit rebellious, even though I did do well in school. And in high school, I had a wonderful chemistry teacher who got me a position at Case Western Medical School. I worked there 3 summers, doing research starting after 11th grade, research on metabolism of red blood cells. So as many of you know, my lab has been working on the development of red cells for many, many years, and I've actually been studying red cells for 60 years. But also working in a lab, particularly as a high school student, gave me an insight as to what research actually could be. But as I said, I was rebellious. So when time came to apply to college, I applied to and got accepted by the one school everyone thought I should go to, which is Harvard. But I had visited Harvard, and I didn't like it. And I had a very good guidance counselor who convinced me to look at small liberal arts colleges. And I visited Oberlin and Kenyon and decided, against everyone's advice, to go to Kenyon. It was then an all-men's school, 400 men, part of the Episcopal Church, but I realized there I could break my own path and really devise my own education. So I was there 3 years. I majored in math and chemistry. I remember the summer before my last year at Kenyon, I worked in Carl Djurassi's lab, the birth control pill developer at Stanford. And when I got back to Cleveland, I visited the medical school, and my mentor, a nephrologist named Robert Eckel, took me to see the dean. And the dean started trying to convince me to go to Case Western Medical School. And I remember going home that evening and telling my parents the good news that the dean wants me to go to medical school and I don't even need to apply. But the bad news is I'm going to graduate school. Okay? So my parents felt I was wasting my life. It got much worse during my first day at graduate school. Okay? So I go to Rockefeller. We drive with my 2 younger brothers in tow, arrive there about 2 o'clock in the afternoon, and I'm escorted into this fabulous single room in the graduate student residence hall. And, you know, beautiful, brand new building, share a bathroom with a neighboring room. The problem was the fellow living in that room was what we used to call a hippie, okay? He did introduce himself. He had long hair, white clothes, sandals, unshaven. The room was a mess, but we don't need to go there. And my mother's parting words were, you know, Harvey, he's not like us. Don't have anything to do with him. Now, 13 years later, when David Baltimore won the Nobel Prize, the situation was completely different. It was my son's best friend had won the Nobel Prize. Okay? So you see where I'm going. Actually, you know, in thinking about it, I realized I shouldn't be too hard on my mother. That fall, I gave her the job of finding me a girlfriend. And, well, we've been married a long time. Anyway, but that is how it started, okay? And a lot happened between the time I started graduate school and these 1970s. I finished at Rockefeller. I was a postdoc with Francis Crick and Sidney Brenner at the LMB in Cambridge. I joined the MIT faculty in 1968, the same time David joined. David, being much older than me, had done several postdocs and had his own lab at the Salk Institute. Whether by design or coincidence, David and I were given adjoining offices in the old building 16. And we shared 3 large laboratories. Our research interests were very different. Mine on translational control of protein synthesis, David on viruses it was soon after he arrived that he discovered reverse transcriptase. And that's another story, which, of course, won him the Nobel. But we realized that we could work together. We trained 3 or 4 graduate students and postdoctoral fellows. For instance, his postdoc, then his wife, Alice Wong, brought vesicular stomatitis virus to the lab. And we started studying, my lab started studying the biogenesis of the VSV glycoprotein, a model for all membrane glycoproteins, the system Jim Rothman used much later to win his own Nobel Prize. So we were collaborating together from the very beginning. And that's one of the lessons that I'll leave for you, the importance of collaborations. David moved with Salvador Luria to start the Cancer Center around 1973, the old Cancer Center on Ames Street. And I was left alone in Building 16. But we still kept in contact. And then Jack Whitehead appeared on the scene. Now, Jack is one of the heroes that I mentioned. He was not a scientist. He was a businessman who understood science. Jack built his father's company, Technicon, into a major producer of devices that measured automatically blood and urine samples, called the Technicon Autoanalyzer, a technology that was actually developed in Cleveland. And in the early 70s, he sold the company to Revlon for several $100 million. And Jack lived in Greenwich, and many of his friends were philanthropists. But Jack wanted to do it differently. Rather than give a few million here and a few million there, he wanted to start his own research building. He didn't quite understand how to do it, and that's another story, his attempts to do it at Duke and Rockefeller. But finally, through Joshua Lederberg, he met David Baltimore. And David was a visionary young scientist. He was probably the most important biological scientist in the United States in the late 70s. He was one of the founders of the Asilomar movement and many other public activities. And David met with Jack and convinced him that the institute that Jack would start, a basic science research institute, had to be part of a major research university, not stuck in Tarrytown next to Technicon. Okay, and what happened then was a then totally new way of governance and organizing institutions. Jack sensibly did not want to give his money to MIT. He did not believe in large institutions. He wanted a small elite research institute. Elite's a word I'm going to come to again and again because that's really the idea. But basically what emerged was highly controversial, was eventually brought to a vote of the MIT faculty, and then approved. So we are chartered as what they call an independent 501c3 research institute. That is our tax status. We are chartered as an independent educational institution. But having said that, all Whitehead PIs are regular MIT faculty members. And by statute, the director of Whitehead must be a professor of biology. So I, like all of my Whitehead colleagues, get no money from MIT. But yet we are obligated to teach and do teach, in most cases, core undergraduate courses at MIT. So for instance, for somewhere around 30 years, I taught the undergraduate course in cell biology. So all of our PhD students and undergraduates are regular MIT students. Our postdocs do not have MIT appointments. They only have Whitehead appointments, as many of you know. I'll talk a little bit later about our fellows program that jump-started the careers of many outstanding biomedical researchers, and I'll mention some of them by name. But also, we wanted to have a culture here of collaboration, and I'll emphasize that later on. That is, the Institute was supposed to be, in quotes, developmental biology, but it basically meant the best people you can find to hire. And collaboration between labs is encouraged by the geography of the building, and I'll come to some of the other facilities. But first, David needed to hire faculty. Now, someone asked who that person is. It's me. This was taken in the 70s in our old lab in Building 16. That's Rex Chisholm to my left and Charles Zucker. But I needed to get out of that ancient lab. But David, among other qualities, had excellent taste for people. And he hired a young Bob Weinberg, okay, as a faculty member. Whoops, what happened? Here we go. There's Bob Weinberg, Jerry Fink. Yeah, we're young ones. Rudy Yanish, a couple of assistant professors, Richard Mulligan, Paul Matsadero. Those are older pictures. Here's one I particularly love, recruiting. Okay. Well, you know, you want a history, you get it. Now, here's something unique, and we often don't talk about it. Jack instituted something absolutely unusual, which is the Whitehead Faculty Retreat. Every year, beginning before the Institute actually existed, he flew all of us out with our families to Vail, Colorado. In fact, the first 2 or 3 years, my wife and I, with our 3 adolescent children, flew on a private plane directly to Eagle Airport. And Jack was a world-class powder skier, and you had to learn to ski. I won't go through all the people there, but that's Jack. That's David Baltimore, Alice, his wife, 2 of my kids, Rudolph, and anyway, you get the idea. Now, we also needed to build a building. And the history of Whitehead is tied in with the history of Kendall Square. This is Kendall Square in 1976. Okay, it was an industrial wasteland, polluted by all kinds of chemicals. This is Main Street here. You can see a few of the buildings, and there was simply nothing here. We were the pioneer of Kendall Square. Our building on polluted soil was done here. It was cleaned up. And the original building was only about 60% the size of the current building. It was designed so that with internal corridors in the labs, you all know this, such that people could move from one laboratory to another and encourage collaboration. We did call ourselves, I know it's unfashionable to say you're elite, but that's what we called ourselves. And Jack called us that. He used to go to our retreats, he used to give lectures, and he used to stand up there and say, "You guys are the greatest. You're doing the greatest research in the world, and you should be the best research institute in the world." I can't imitate him, maybe one of you guys can. But you get the idea that we really felt we were going to be, those are some of my faculty colleagues there. Anyway, we built the building and populated it. And, you know, I want to acknowledge some of the other people who were there at the beginning, particularly John Pratt, Margaret Sand, Marilyn Smith, who was David's assistant, in really getting the Institute started. Now, I remember very clearly the day we moved into the Institute from across the street. It took us 3 days. We had a moving van, but a lot of the lab stuff we had to move ourselves. I had to move my own office. And I told the people in my lab, look, if we are all moved in and everything's clean and we're ready to go on Thursday, we'll have a party at our house and we'll have takeout Chinese food. And we had just moved into a large house on Fisher Avenue in Brookline, and then started a tradition which lasts to the present day, because we had just moved into the house, and the house had a swimming pool in the back. And someone asked me, had I ever been in the swimming pool? And I said no. And it was decided that I should go into the swimming pool. And every year now, we have this party at my lab. And this is actually our condo swimming pool, but it becomes the centerpiece of a party, and people simply come to watch me get thrown into the pool. So I'm not sure whether it's good or bad, but in all honesty, I have to tell you about this. Also, as I said, we constructed the building to encourage collaboration. And one of the very unusual things we put in the building was a cafeteria. You may not realize it, but back in 1983, when the building was built, research buildings did not have cafeterias. But we, the founders, together with our architectural advisors, felt that having a place where people could come and meet and eat and talk would be essential to create this culture that we wanted to do. And as you know, the cafeteria has found many, many uses. Here's one I'm not sure you knew. It was the only wedding that I know of at Whitehead. It was actually a lovely wedding of 2 graduate students, Huang Ming and Miao Qing, and they asked me to officiate, which I did legally. Okay? Yeah, it was a legal wedding. If you didn't know it, it's kind of fun, because you have to go to the State House, but you need to have a letter from someone in authority that says you're of good moral character. So on a certain day, I went to Susan Toccio, David Page's assistant. I said, Susan, don't ask questions, but I need a letter from David. Okay? Anyway, and they're still happily married. And another thing that we put in the Institute is faculty offices that were large enough to house small meetings. And again, my office has housed a whole variety of meetings. Here's probably the most unusual one, because the bride needed a place to get dressed for the wedding. And, but it worked. Okay. Anyway, to continue the Whitehead saga, we hired a number of faculty. They came, as is our tradition, as junior faculty. That's Terry, of course, and Hazel. And as I did, and virtually all of us did, grew up in the MIT system and became senior professors. The Whitehead Fellows Program was one of the signature programs, and it was David Baltimore's vision again. And this is the concept of taking young scientists directly out of an MD or PhD, scientists who have both the scientific and personal maturity to run their own laboratories at a young age, and give them the resources to do innovative and creative science. And one of the first was Peter Kim, who became faculty. David, I don't have a picture of you at that age, but David came very early as well as a faculty, also became faculty. And then Eric Lander, of course, who was a mathematician teaching at Harvard Business School. If you didn't know it, Eric never took a biology course, but he had a vision as a young man of mapping the human genome, and we felt he would be a very good fellow at Whitehead because all he needed was a room of computers. I'm serious. Okay? Anyway, that was the 80s. David left Whitehead in '89 to become president of the Rockefeller University. That actually was not a successful presidency. And he returned to MIT after about 2 years. Not to Whitehead, but to MIT. After a few more years, when the conflicts resolved themselves, he was appointed president of Caltech, and then moved to Caltech, where he is today. But another of our visionary leaders became director then. You recognize him? Jerry Fink, okay? And Jack Whitehead. And Jerry did two important things. One is he raised considerable funds of money to expand the building. What he realized is that the original building was too small, that biology was incorporating lots of new techniques, microscopes, computers, and so forth. We needed extra space and had the addition built. And it was one of the more moving events. Paul Tsongas, none of you would know, he was a senator and for a brief period of time a presidential candidate. And he was dying of a blood cancer and literally got out of his deathbed to come to speak at the dedication of the new building. And he commented how important the work we were doing here was, not that it was going to lead to a cure for him, but eventual cures to someone else. So Jerry is credited with the building and also helping forward the Human Genome Project. Because in the 80s, late 80s, early 90s, Eric Lander began proposing the heretical notion that one should sequence the entirety of the human genome. And that was not a common thought at the time. Many of us, I put myself in that group, had really little idea what sequencing the human genome meant, all 3 billion base pairs. Jerry and the board, if I understand it, invested somewhere north of $50 million in sequencing machines and rented space to allow Eric to get started on this public sequencing of the genome. There was a fear that a private company, which was racing in parallel to sequence the genome, might literally patent the human genome. Eric he really led a large number of people in the public genome sequencing, which eventually was successful. When was it? 2001, 2002. And so that was done, if you didn't know it, at the Whitehead Institute. It was done in outbuildings that we had largely rented. Eric, in turn, was another of these visionaries who saw the need for an institute, a separate institute, that would build on the human genome sequence and use it to understand and eventually treat a whole variety of human diseases, understand a whole set of organisms by getting their sequence, and so forth. And he recruited this fellow, Eli Broad. Eli was he's still very much alive. Eli is a Los Angeles businessman. He, of course, built or paid for the new, the museum in Los Angeles, the Symphony Hall in Los Angeles. And Eric convinced him to give quite a lot of money, several $100 million, towards the development of a separate research institute, a daughter, as it were, of Whitehead, next door, the Broad Institute, that is now something that has enhanced DNA biology, RNA biology in every institution in Boston in the United States. Now, in parallel, the human genome catalyzed the formation of a number of companies. There were biotech companies in Kendall Square before that: Biogen, Genzyme, Repligen, a few others. Companies that were using the technology of the 80s to develop recombinant proteins. But the human genome launched, or was a major part in launching, a number of biotech companies, many of the hundreds that we now have around Kendall Square. And you can see the striking difference. Almost every building you see in the center has something to do with biomedical research. And what is now called Kendall Square used to be called Genetown, because it was where the genome was sequenced. So I don't think Whitehead can take full credit for it, but we can certainly take a lot of credit for the development of Kendall Square. And in a moment, I'll share with you some of the companies that came out of Whitehead, some of the many companies that came out of Whitehead. Jerry Fink, as I mentioned, was our second director. We were privileged to have, and still have, great directors of Whitehead who lead us forward. Sue Lindquist came in 2001, served as director for about 3 years, stepped down, but then wisely stayed at Whitehead as a faculty member doing her fabulous work on protein folding and neurodegenerative diseases. And David, that's you, took over. Yeah, it's pretty good. David took over in 2004 and has hired helped hire or hired many of the young, outstanding young faculty that we have here today, and is really driving the next generation of Whitehead leaders going forward, because some of us are getting a little old. Anyway, that's Bob Weinberg. We celebrated our 25th anniversary in 2007. This is a celebration in front, but we had a dinner. I couldn't find pictures of it at the MFA, and among the speakers were our two senators, John Kerry and Ted Kennedy, and our governor, Deval Patrick, and lots of stuff happened. Now, when I gave the talk to the potential donors, I made lists of accomplishments of the Whitehead faculty. And I'm going to share those. I've updated it. I'm just talking about the senior faculty, the ones who have or got tenure. But just to go through so you can see what accomplishments were really made. And these are cherry-picked, so there are many others. Obviously, the first human oncogene, the model of chronic myelogenous leukemia that led to the first drug for leukemia, imatinib. Richard Mulligan developed some of the first gene therapy vectors. My lab cloned a number of receptors and transport proteins. Jerry, among other things, analyzed the weird genetics of this important fungal pathogen, Candida albicans. Rudolph, one of the first, actually, the discoverer of epigenetics, and using gene cloning in stem cells to correct a genetic defect in mice, presaging gene therapy, understanding how viruses evade the immune system. I'm going on. MicroRNAs as key regulators of gene expression, Dave Bartel. Physical map DNA sequence of the Y chromosome. Paul Matsudera's work on actin cytoskeletal proteins. Eric sequenced the human genome. Susan Lindquist using yeast to study neurodegenerative disorders. Terry's beautiful work on regulation of DNA replication. Richard Mulligan, I have him twice actually. Anyway, that's okay. Peter Kim, understanding the mechanism of HIV infection. Rick Young pioneered DNA chips to discover regulatory elements, super enhancers. Hazel's work on the development of the brain and proteins required for brain development. Ian's central work on the centrosome and chromatin replication. Planarial stem cells by Peter. Really discovery and elucidation of the mTOR pathway by David Sabatini. And since none of them really worked on plants, we have two great plant biologists here, Mary and Jinky. And if you haven't been in the greenhouse, it's kind of cool. I also have 3 slides that point out some of the recognitions of the faculty. We have 4 HHMI investigators, and if you add Susan, who was one, it would be 5. Susan, like Bob and Rudolph, received the National Medal of Science. It's only given to 2 or 3 life scientists every year, so it really is remarkable. And over half of the Whitehead faculty are members of the National Academy. This number is higher than most states, okay? It really is remarkable to have this many leading scientists, and obviously we have younger ones who are going to get there. So it really is an incredible concentration of people. I mentioned the Whitehead Fellows Program. I wish I could take the time to enumerate all of the 32 fellows, 3 of whom are still at Whitehead. Every one of them has gone on to stellar academic careers, and as you can see, 7 have been elected to the National Academy. Not that it's the only measure of accomplishment, but it certainly is a major measure, and that's quite incredible. And all of them have gone on to amazing careers. We even have an astronaut, okay? And who's spoken here several times. And again, we are in the business of training people, and we don't often articulate it enough. Every lab can show a picture like this of alumni who dominate meetings in their own field. And most of the people in this big picture are faculty. These are people from my own labs at some of the top universities and research institutes in this country and abroad. I won't name them all, but any of the, particularly the older labs at the Institute could do this. And in my case, I've trained a lot of alumni. Here are the 2 that went on to win Nobel Prizes. Aaron Ciechanover, who was of course the discoverer of ubiquitin, he actually did this before he came to my lab, but worked on it while he was a postdoc. And then Jim Rothman, who brilliantly took the VSV glycoprotein biogenesis system that we had studied and used it to elucidate proteins in vesicular transport. And this was the Nobel Prize he shared with Randy Sheckman and Tom Sudhoff. I mentioned collaboration, and collaboration is key. Whoa, what happened? Don't send. Don't worry, it'll come back up. Here we go. Hey, look at that. I just put in 4 slides to illustrate how my own lab's research was dependent on collaborations. As I mentioned, a major part of what we've done for the past 35 years is study the details of how red cells are formed. Here, a study of knockout mice was done together with Rudy Yanish. Dave Bartel pioneered the discovery of microRNAs in mammals. And it was a postdoc in my lab, Shengzhen Chen, who worked with a student in David's lab and David to really the first demonstration that microRNAs played a key role, lineage-specific microRNAs, played a key role in mammalian development. A collaboration between a postdoc in my lab and Andrew Steele, who was in Susan's lab, led to several papers elucidating one of Susan's favorite topics, prion proteins, showing how important they were in the function of stem cells. And then finally, work that we did together with Rick Young's group, looking at chromatin modifications and their role in gene expression during red cell development. Again, examples, some of many, that any of our labs could do to illustrate how important this is. Finally, we have other things that encourage collaborations. I think everyone loves the retreats in New Hampshire. You may not realize not every institution does it. It costs a lot of money. And I'm sure David, if I asked him, I've never asked him, but that the money is worthwhile because of the collaborations, interactions, grants that come out of these interactions that almost all of you have seen during the Whitehead retreats. We also do outreach at Whitehead. I was involved in setting up this program many, many years ago, where we bring in over the April school vacation week high school students for lectures, lab demonstrations, and tours of companies. We have several programs for middle school students. I can tell you our grandson Isaac took one of the programs, and I remember driving him home one day, and he was telling me how great it was to see the decaffeinated coffee plants growing in Jinky's greenhouse. It was quite amazing. So the middle school program is, I think, one of our great successes. We have our own child care center. And that was the gift of Britt Darbeloff, and I think also indicates that we, the whole group collectively, feel how important it is for people to feel comfortable with families and to get the kind of outside support that they need in order to do good research. A cynic could say, yeah, we have a child center so that people can work in the lab 24/7. And that is partly true. I'm almost done. I just want to end with, the impact Whitehead has had on the broader biomedical community, particularly in companies. As Claire said in her kind introduction, I've been involved in starting a number of companies, including 2 very large ones in the area, Genzyme, which is now part of Sanofi, and Millennium. But many of the Whitehead faculty have started public companies. These are just the ones that we could find in the past 10 years. And these employ many people, I don't know how many, And many of these companies have or will soon have drugs on the market for currently untreatable diseases. And this still goes on. That is, many of us over many years have started companies. These are just the private companies, and I'm sure some of them will go public in the near future. Here's a pretty picture of it. And this just illustrates, and this is an important slide in terms of geography. It illustrates the central nature of Kendall Square in an ecosystem of biotech companies, venture capital firms, incubators, and so forth. By way of illustration, when Hidde Ploch and I started Rubius out of technology in our laboratories, we worked with Flagship Ventures, whose office then was a 5-minute walk from Whitehead. Our first labs were in Lab Central, which are just up the street another 5 minutes. And that was important because we could walk and check things out. When we got too big for Lab Central, we moved into this building, which is an old Pfizer building, because Pfizer was consolidating all of their research from Connecticut somewhere up where the Lab Central sign is. And right now, Rubius is in a considerably larger space there. The point is, what's grown up around Whitehead is a huge network of startup, middle-sized, and large pharmaceutical companies. Let me end with just a few general comments. One, and I think everyone needs to be aware of it, is the changing pattern of support of biomedical research. And I mentioned philanthropy several times, and I'm going to reiterate it now. This is the distribution of funds that support all of us at Whitehead. Okay? 20 years ago, 30 years ago, 2/3 of the support was from federal grants. That is now 1/3. Okay? This is true of Whitehead. I know it's true of Boston Children's Hospital. It is true of the Dana-Farber Cancer Center. It is true of every major research institution. And this is a huge threat to the continued growth, not just of Whitehead or Children's Hospital, but of the American biomedical research enterprise. And much of the difference is made up by income from the endowment, which is currently north of $400 million, and then other funds, including foundation and corporate philanthropy, individual philanthropy, and so forth. So going forward is not just for Whitehead, but for all American research institutes, particularly even the top ones. The funding is going to be a difficult issue. But we continue. We still have Whitehead faculty retreats. Here you can see the one a couple of years ago. And really, I'm going to end where I started with is the founding of the Whitehead Institute. Here we are all together. David, somehow you escaped. I couldn't find you. But the others are all in this picture. I think you can probably recognize them. But this was celebrating David's 80th birthday in Pasadena. Anyway, I'm done. If you have specific questions, I'll do my best to answer them. Thank you.